Hi, I'm Michael Doyle. I'm Director of the Center for Food Safety at the University of Georgia. And today I'm going to talk about some innovative chemical treatments that can be useful to kill foodborne pathogens. There has been a consumer movement that initially started in Europe and has spread to the United States to remove food additives or chemicals from foods that are largely used to preserve foods or to color foods or for other reasons. I describe this as a chemophobia and the movement then towards natural foods. And this movement has been largely created and expanded by what I call the blogosphere. That is, people who are using the internet to communicate, and sometimes, in fact many times, there's misinformation that's disseminated by this route, and it's not well grounded in science. There's a well-known blogger here in the U.S. named Marion Nestle, who is with New York University. She's a nutritionist, but she's one of the most widely read food safety bloggers here in the U.S. And she has a movement to reduce consumption of what she calls processed foods. And Marion is a very smart woman. I think she's a very good nutritionist, but her concept of processed foods is different than what I would consider a processed food. She defines a processed food as the number of food additives that are present in a food, especially those that have esoteric or hard to understand names. And so she is part of this movement to reduce the use of food chemicals. That can have significant food safety ramifications because many chemicals that are added to foods are added with the sole purpose of preserving the food or killing harmful microbes should they be present. A good example being benzoic acid or sorbic acid, which is added not only to kill or control the growth of molds and yeast, but can be useful to kill salmonella or staphylococcus aureus, and in the case of sorbate, even Clostridium botulinum. And so what I'm going to talk about today is what I would describe as innovative chemicals that are more natural-like, that could be used to kill harmful microbes that you can find in foods. And this approach, I hope, could be at least a happy medium between scientists who want to have some form of protection added to foods and some of the bloggers who are more interested in having natural foods. So I want to talk about several approaches that might be considered as alternatives to some of the present-day preservatives that might be used in foods. And the first I'm going to describe is something called levulinic acid in combination with a chemical called sodium dodecyl sulfate or sodium lauryl sulfate. Secondly, we'll talk about spices and some of the antimicrobial effects of spices. Thirdly, talk about cranberries. In fact, there's a variety of berries that have naturally occurring benzoic acid. This is the chemical that some bloggers want to remove from foods, but actually there are many foods that naturally contain benzoic acid and perhaps could be added naturally to transmit this chemical to foods. Touch upon lactoferrin. This is a chemical that's naturally occurring in milk. And then talk about the concept of natural antimicrobials. And I have it in quotes because this is a way of producing antimicrobials, perhaps by fermentation, and adding this fermentate to foods, which is often done here in the United States in the form of a preservative for bread. It's a fermented whey product in which Propionibacterium bacterium is fermented in this whey, and then the whey is added as fermented whey. That's how it's labeled on the ingredient panel. So first, let's talk about levulinic acid. This is a somewhat unique acid. It can be prepared by treating biomass, such as waste from your yard or produce waste. Treat it with acid, typically sulfuric acid, heat it to 160 to 200 degrees centigrade, and what remains after that treatment is largely levulinic acid. As I mentioned, the feedstocks for making this can include vegetable waste, include wood waste, paper mill sludge, or even municipal solid waste. So it's in my view, a very natural and sustainable approach to preparing this acid. Studies have been done to show that it's not a toxic acid. The LD50 oral dose in rats of acetic acid is about 3,000 milligrams per kilogram. And the dose for this type of acid, levulinic acid, is about 1,850 milligrams per kilogram in rats. So it's not too much different than acetic acid. This levulinic acid has been used in cigarettes for many years. That's why it's been so widely studied in determining that it's not toxic. But it does increase nicotine delivery in smoke and binding the nicotine to neural receptors in the brain, and that's the primary reason for which it's added to cigarettes. Levulinic acid is also used in reconstituted vegetables, in ice cream, in baked goods, in fats and oils, in candy, in cheese, in beverages, and these types of foods here in the U.S. It's GRAS approved by the FDA as a flavor adjunct. The other component is sodium dodecyl sulfate, which is also known as sodium lauryl sulfate. It's synthesized by reacting lauryl alcohol with sulfuric acid and neutralizing this with sodium carbonate. And it's considered to be an anionic detergent. It's used in toothpastes at levels of 0.5 to 2%. It is generally recognized as safe as a food additive. It's used as an emulsifier, a surfactant, and a thickener in foods. It's used in dried egg products at 0.5% as a whipping aid. It's used in egg whites as an emulsifier, and it's used in fumaric acid acidulated fruit juices at levels of 25 parts per million as a surfactant. We have done studies to define the antimicrobial efficacy of the levulinic acid and SDS alone and in combination. And initially these studies were just done in pure culture with salmonella at room temperature. And some of those results are showing in this slide. You can see that when we just have a salmonella control for 30 minutes, it doesn't die off. It stays at the same level of 7 logs. And if you apply levulinic acid up to 2%, after 30 minutes, there's a slight reduction, less than a 1 log reduction at 30 minutes, essentially no reduction after 10 minutes. And if you increase the levulinic acid concentration to 3%, you might get slightly less than a half log reduction in 10 minutes and a greater reduction after 30 minutes. But in that relatively long exposure of 10 minutes, there's very little reduction. And in comparing this to acids like acetic acid or lactic acid at 0.5%, you can see that lactic acid tends to be a little bit more effective after 10 minutes in reducing salmonella, maybe about a log reduction, whereas acetic acid had very little effect after even 30 minutes in reducing salmonella. When we test SDS, the sodium dodecyl sulfate, at 0.05% up to 2%, we do see little to no reduction at 0.05%, and we get up to 2%. There is considerable reduction after 2 minutes, even after an immediate response after 15 seconds, which is what it would take to manipulate the culture and enumerate the salmonella. We get about a 4 or 5 log reduction. Now, when we add these two chemicals together, the levulinic acid with the SDS, and we're only using very low levels of SDS, 0.05%, you can see that we get complete inactivation of salmonella, a 7 log reduction when we're using 0.3%, 0.5% lactic acid with 0.05% SDS. It's not unique to levulinic acid. When we use other acids like caprylic acid or lactic acid, we get similar responses. It's the synergistic action of the SDS with the acid that kills the salmonella. Now, we've applied this treatment, largely SDS plus levulinic acid, in a variety of conditions that relate to food treatments. And I'm only going to give you a few examples, starting out with treating pre-harvest and post-harvest poultry with the levulinic acid and SDS. And the objective of this particular study was to see if we could get a 5 log reduction, that's a 1,000-fold reduction, in a biofilm, as well as on whole chickens, on poultry, transportation cages, and on processed poultry within 30 minutes. So the other goal is to determine if we can inactivate salmonella by this levulinic acid SDS treatment in the presence of what we call organic load, such as chicken feces or chicken feathers being present. So here are some results of these studies. We find that when we just have salmonella present in water that contains chicken feathers at 1 part chicken feathers to 100 parts water, we didn't get any reduction in salmonella after 30 minutes. When we had 1% levulinic acid and 0.1% sodium dodecyl sulfate, within 2 minutes we had complete elimination of the salmonella, so it was greater than a 7 log reduction. With the feces, when we had 1 part feces to 20 parts water, again, we didn't get any inactivation of the salmonella within 30 minutes, but when we added 3% levulinic acid and 2% SDS, within 2 minutes, again, we had complete elimination of the salmonella. We've done these studies also on poultry wings and applied different levels of levulinic acid and SDS, and we find that one of the better treatments that still leaves the poultry tissue intact in looking like it's fresh like poultry skin was the treatment with 2% levulinic acid and 1% SDS. And within 5 minutes of this type of treatment, and this treatment, by the way, was done at 8 degrees centigrade. This is a colder temperature than the room temperature studies that were done as I previously described. But we found about a 3.5 log reduction when we used this treatment at 8 degrees centigrade. Another important finding is that the treatment solution itself, that is the levulinic acid SDS solution that was the wash water, if you will, did not become highly contaminated. We did find surviving salmonella, but only by enrichment in this treatment solution. So there was more than a 7-log reduction in the treatment solution during this 5-minute treatment. When we increased the levulinic acid concentration to 3% and had 2% SDS, we were able to, within 2 minutes, eliminate almost 7 logs of salmonella, and we did not find salmonella in the treatment solution. Again, this study was done at a refrigerated temperature, about 8 degrees centigrade, and the tissue itself, that is the poultry wings, still appeared to be white and fresh-like, so it didn't adversely affect the color of the tissue. We've also looked at biofilms, and we grew salmonella as a biofilm on several different types of matrices such as stainless steel and polyvinyl chloride, on rubber, on glass, even on polyethylene, and then treated these biofilms on these different coupons with 3% levulinic acid plus 2% sodium dodecyl sulfate, and the sulfate, and the pH was 2.8. And the application of the levulinic acid plus SDS was done as a foam. When you pump this material up and spray it on the surface, it comes out as a foam, and it stays foamy for at least 20 to 30 minutes. And you can see after 20 minutes of exposure of these biofilms on these different matrices, we were able to get a 5 to 7 log reduction of the salmonella. Didn't totally eliminate it, but we went from 7 to 8 logs down to 1 to 2 logs. So that was a major reduction for something that's entrapped by this polysaccharide material in a biofilm. We then took this to the field, took this concept to the field, and applied it to poultry cages. These poultry cages were contaminated, if you will, with feces from birds, chickens that were brought to the processing plant, and we didn't eliminate all the contamination or the feces on the cages. We simply sprayed the cages, and the part that we tested were the doors on the cages. And we sprayed 3% levulinic acid plus 2% SDS as a foam, room temperature, on 100 different transport cages, and held this for 45 minutes. And the entire surface of the metal cage and door was covered with this foam. We then took a sponge and tested a 5 centimeter by 5 centimeter surface of the cage door after the foam had dissipated after 45 minutes, and we tested it for salmonella and coliforms and aerobic plate counts. And the results of this study are shown in this slide. You can see that 19% of the cages were contaminated with salmonella before the treatment. After the treatment, only 1% of the salmonella was present on the cage doors. The coliform counts went from 6.8 on average logs per 25 centimeters, down 25 square centimeters, down to 1.2 log colony forming units per 25 square centimeters. The aerobic plate count went from 8 logs down to 4.8. So we had a substantial reduction in salmonella contamination, as well as coliform counts and aerobic plate counts. We also, in this poultry facility, sprayed the area, we call it the receiving area, where the cages come into the poultry slaughterhouse, and we sprayed the floor with the 3% levulinic acid plus 2% SDS as a foam, room temperature, held it for 45 minutes, and then we did a swab sample of a 10-foot by 10-foot section of the floor. And I'm sorry, that's the area that we sprayed in the receiving area. Then we did a swab sample of a 5 centimeter by 5 centimeter surface in 50 different locations on that treated floor. And these are the results of our tests. Before the treatment, we didn't find any salmonella in any of the 50 samples, and we didn't after the treatment either, but the mean coliform count was almost 8 logs per 25 square centimeters. And after the treatment, it was less than 1 log. So we had almost a 7 log reduction of coliforms. And with the aerobic plate counts, we went from 8.4 log per 25 square centimeters down to 3.6 logs. And so that's almost a 5 log reduction. Again, a very significant reduction of contamination. Another approach that we have used, not with poultry, but in this case with cantaloupes. This is a melon that we've had issues with listeria and salmonella contamination. Several outbreaks have been associated with salmonella on cantaloupes, and a major outbreak in the U.S. associated with listeria, where 33 people died from eating listeria-contaminated cantaloupes. And so our goal was to determine if we could use this levulinic acid plus SDS treatment to substantially reduce salmonella contamination should it be present on cantaloupe. And we tested 2 areas. We tested the outer surface of the cantaloupe as well as what we call the stem scar. It's like where the umbilical cord is for the cantaloupe attached to this umbilical cord that goes to the plant in the ground. In any event, the stem scar is an area that if the pathogen is present, it's very hard to remove or eliminate, and that's an area that the organism can penetrate into the tissue of the melon. So that was the reasons for testing both the outer surface, we call it the netted rind, of the melon, as well as the stem scar. And when we started the study, just the control, we had about 7 logs of salmonella on the stem scar and about 6 logs of salmonella per gram on the netted rind. We applied a water treatment, and that reduced the salmonella contamination on the stem scar by about 1 log, and on the netted rind by about 1.5 logs. We used 120 parts per million chlorine, which is a typical treatment that's used in these tanks, these dump tanks that are used to wash the cantaloupe in many facilities. And with that treatment, we saw about a 1.7 log reduction per gram on the stem scar and about a 2.5 log reduction per gram on the outside surface of the netted rind. When we used the 2% levulinic acid and 0.2% sodium dodecyl sulfate treatment, we had about a 1.5 log reduction on the stem scar, not much different than chlorine, but we had a much greater reduction on the overall surface of the melon. It was about a 4.5 log reduction, which was substantially more. It's very difficult to reduce the pathogen load on the stem scar once it gets attached to that. And so a recommendation that we've made is that if you want to cut off that stem scar before cutting the melon and eating the internal content. We also looked at the cantaloupe, or the melon flesh, the meat that's inside, 18 hours after we had inoculated the cantaloupe with a high dose of salmonella, and we treated the cantaloupe with water and chlorine and with the levulinic acid and SDS, And shown here are the results of that study. With the control at the stem scar, 5 of 12 cantaloupe samples were positive for the salmonella in the flesh underneath the stem scar. In the flesh of the cantaloupe underneath the netted rind, the control had 5 out of 12 positives for salmonella. When we used water to treat the cantaloupe, all 10 of the 10 melons were positive for salmonella within the flesh at the stem scar, as well as the flesh of the netted rind. When we used 120 parts per million chlorine, 7 of 10 of the flesh at the stem scar was positive for salmonella, and 6 of 10 of the flesh of the netted rind was positive. And when we used the levulinic acid SDS treatment, 14 of the 20 stem scar samples, flesh samples, were positive. Again, very difficult to remove or eliminate the contamination at the stem scar. But when we tested the rest of the melon, the flesh of the melon under the netted rind, we found none of the 20 melon samples contained salmonella. So it was this treatment of the levulinic acid SDS was very good at removing and killing the salmonella on the surface of the melon, but not at the stem scar. Another group of researchers had published some recent results in the Journal of Food Protection where they applied bacteriophage at levels of 10 to the 8th plaque-forming units per gram and used our 0.5% levulinic acid plus 0.05% SDS treatment to inactivate salmonella on cantaloupe, and their goal was to focus on the wash water, the water that was used to wash the cantaloupe. And when they used just tap water, no treatment, they found that the water contained about 6 logs of salmonella. That was their inoculum, 6 logs. And then when they used bacteriophage, they found that they had about a 1 log reduction. And this was applied to the cantaloupe. The cantaloupe was about 10 degrees centigrade. It was applied 24 hours before a wash treatment. And so they saw about a little over a 1 log reduction with just bacteriophage. When you tried the levulinic acid and the SDS, after a 1 or 5 minute treatment, there was about a little over 2 log reduction in the potable water, and that's in the challenge water. There was about a 4 log reduction. And when you applied the levulinic acid SDS with the bacteriophage, you got about a 4 log reduction in the potable water, and an even greater reduction in what we call the challenge water. And if you used 200 parts per million chlorine, the level of reduction was similar to what we saw with just the levulinic acid, that is in the potable water. But when you used it in the challenge water, and when I say the challenge water, this is the potable water that has all these dissolved solids and organic carbon that helps neutralize chlorine in particular, but many disinfectants are sensitive to this organic load. So that's the difference between the potable water and the challenge water. So basically, there was very little reduction of salmonella in the 200 parts per million free chlorine treated challenge water because of the neutralization effect of the organic load. And we didn't see that with the levulinate acid in the SDS or the levulinate acid plus the bacteriophage. But the point is, the levulinate acid plus SDS worked much better with or without organic load, and adding the bacteriophage had an additive effect on killing the salmonella. So here are some advantages of using levulinate acid plus this SDS treatment, and it largely depends on what concentration is applied, but it has very strong antimicrobial activity against salmonella, E. coli 0157, and Listeria monocytogenes. You can get up to a 5 to 7 log reduction of these pathogens depending on the concentration and the temperature that's used. The antimicrobial activity of this treatment is not readily inactivated by the organic load that's present. The beauty of levulinate acid compared to other acids like other organic acids such as acetic acid or lactic acid is that it's not corrosive. That you can treat stainless steel with these high concentrations of 3% levulinate acid and it doesn't corrode the material, whereas lactic acid and acetic acid and peracetic acid are all corrosive, and with time it will have an adverse effect on the surface of the equipment. It's also, levulinate acid is not irritating to skin. The levulinate acid plus SDS treatment removes biofilms and kills bacteria in biofilms. It does have foaming capacity. If you want that, under pressure you can apply this material and it will foam up. You can also add an anti-foam so you don't get foam. And that foam will last at least 20 to 45 minutes depending on the temperature and other conditions. Also, another important aspect is, and this is how levulinate acid differs from a lot of other organic acids, is that the food retains its organoleptic characteristics, its color, its appearance, texture, odor. If you add, for example, acetic acid or lactic acid at 3% to certain types of lettuce or fresh produce, it'll often brown and wilt readily. That doesn't happen with the levulinate acid treatment. In poultry, we see that when you treat with levulinate acid and SDS, it retains its sensory characteristics, its color and appearance throughout shelf life of the product, the normal shelf life. There are some disadvantages, however, in applying the lactic acid and levulinate acid treatment. When you use frozen tissue, it's not effective. You have to have a higher temperature so that the levulinate acid SDS can be effective. Typically we find that anything below 8 degrees centigrade greatly reduces the activity, the antimicrobial activity of the SDS and levulinate acid. And it can foam very excessively if you put this material in a highly aerated condition, such as a chicken scalder, and then you need to apply a defoaming agent to mitigate that problem. I share with you here a list of a few references. We have many others that relate to results of treatments in applying levulinate acid and SDS to everything from poultry to produce to alfalfa seeds, chicken cages, biofilms, viruses, foodborne viruses, cantaloupes, and so on. Next, I want to talk about spices and their antimicrobial activity. And we found, not necessarily our lab, we verified some of these findings, but many researchers have found that a variety of spices naturally contain antimicrobial activity. Cloves, oregano, mustard, garlic, cinnamon, onion, sage, and thyme are among those that I think are among the best of spices that have antimicrobial activity. And there are different types of antimicrobial components in these spices. For example, there's thymol in oregano, thyme and sage. There's isothiocyanate, or at least the precursor to this, in mustard. And when you grind mustard, the enzymes in mustard create this isothiocyanate, which has very strong antimicrobial activity. There's eugenol in cloves and cinnamon in sage. There's allicin, another antimicrobial that is present in garlic. And there's carvacrol in oregano, as well as thymol, which adds to the antimicrobial activity. Oregano oil is a very potent antimicrobial. It contains phenols, carvacrol, as well as thymol. It can inactivate Campylobacter and E. coli and Listeria and Staphylococci and a variety of spoilage microorganisms such as Pseudomonas. 750 parts per million of whole oregano extract can suppress Listeria, but you need the addition of some other things like 2% sodium lactate to reduce the growth by about a half a log of the Listeria in ground beef. And this is after a whole 10 days at 4 degrees centigrade. So it can suppress Listeria growth under the right conditions with the right additives. I share with you results of a study that was done to address the growth of Listeria monocytogenes in cooked ground beef at refrigeration temperature, 4 degrees centigrade, when oregano is added, or oregano with or without cranberries or lactic acid. And it's a busy slide, but the control is at the top, and you can see that Listeria grew from about 3.5 logs to over 5 logs in 10 days at refrigeration temperature when there were no additives present. The bottom curve, the dotted line, is a combination of 750 parts per million of the oregano with and cranberries with 2% sodium lactate or lactic acid, and you can see that there was a beneficial effect. Initially, after 5 days, there was a decrease, slight decrease, a few tenths of a log decrease in the number of Listeria monocytogenes per gram of cooked ground beef. And then after 10 days, the level of Listeria was about a little over a log less than the control. So it didn't totally eliminate the growth of this combination of oregano and lactate. Did not totally eliminate the growth of Listeria, but it suppressed its growth. It reduced it. This slide represents the results of a study with Listeria on beef slices. pH was 6. Again at 4 degrees in the presence of oregano with and without cranberries or the lactic acid. And you can see again that there was a difference between the treatment and the control being the top line, in which Listeria grew within 8 days from about a 3 log level of contamination to almost a 5 log level of contamination. When you apply the oregano plus the cranberries plus lactic acid, within 2 days, Listeria was essentially eliminated to undetectable levels, went from 3 logs to undetectable levels and never returned. And when just the oregano plus the cranberry mixture without the lactic acid was applied, there was a 3 log reduction by 8 days, but it was a gradual reduction up to that point. Next, I want to talk about alloisothiocyanate, and this is responsible for the pungent taste that you have when you eat mustard or horseradish or wasabi. This chemical is slightly soluble in water, and it comes largely from mustard seeds. There's an enzyme called myrosinase that's released in mustard seeds when the seeds are broken or mashed, and this then acts on a glycinolate, which ultimately gives the alloisothiocyanate that is the antimicrobial that's present. And there have been several studies done in using alloisothiocyanate to reduce pathogen contamination in foods, and I share with you some results with E. coli 0157 on beef under different treatment conditions in ground beef, using mustard flour, 10% mustard flour. It's mixed with the ground beef, packaged under nitrogen, stored at 4 degrees centigrade for 21 days. Almost a 4-log reduction of E. coli 0157 per gram of meat. In finely chopped beef, when the alloisothiocyanate was microencapsulated in a gum, and then added to samples that were packaged under nitrogen, stored at 4 degrees centigrade for up to 18 days, there was about a 3-log reduction in 9 days and almost over an 8-log reduction in 18 days. Fermented dry sausages, 500 parts per million alloisothiocyanate, again microencapsulated, was added to sausage, to the batter, was then fermented, and after 28 days there was a 4.75 log reduction. Next, I want to talk about cranberries. And cranberries by themselves contain benzoic acid, naturally containing benzoic acid at levels of about 0.03 to almost 0.1%. Average is about 0.7% benzoic acid. It also has tannins, and these can prevent the tannins can prevent E. coli from adhering to the human urinary tract. And so cranberries, there have been studies to suggest that eating cranberries can help reduce urinary tract infections because these tannins can help prevent the adherence of E. coli to urinary tract cells. But the main reason I think cranberries are useful is because of the presence of the benzoic acid. There are other types of produce, berries, that can contain benzoic acid besides cranberries, cloudberries, mulberries. These have high levels of benzoic acid. And cinnamon, cloves, and prunes can even have some low levels of benzoic acid. When using benzoic acid or cranberries as the vehicle, the antimicrobial activity is going to be largely dependent on the pH. So the lower the pH, the more antimicrobial the activity of the cranberry concentrate. I share with you the results of a study that was done with cranberry concentrate in ground beef under different treatment conditions. The ground beef was mixed with 10% cranberry concentrate, held at either 7 or 21 degrees centigrade. And after 7 days, at 7 degrees centigrade, there was about a 4-log reduction of E. coli 0157 per gram. At 21 degrees centigrade, room temperature, but that's obviously the ground beef is going to be probably spoiled by then because of the growth of the spoilage bacteria. But they did observe an 8-log reduction of E. coli 0157 after 7 days at 21 degrees centigrade. Brief comments about lactoferrin. It's an iron-binding glycoprotein that's present in milk. By using pepsin digestion of bovine lactoferrin, you can generate about a 25 amino acid peptide, which is known as lactoferrin B, and this can inactivate foodborne pathogens. USDA has in 2002 approved the use of this activated lactoferrin for use in fresh ground beef or fresh beef in general. The activated lactoferrin affects pathogens by detaching the pathogens that are already attached to meat. It can prevent pathogens from adhering to the meat, and it even can inhibit pathogen growth. One of my colleagues, Dr. Kumar Venkit, did do a study with lactoferrin B on E. coli 157 in ground beef, and this was actually he did it both in the beef and in peptone water, just 1% peptone water, to see what kind of results could be obtained. The studies in peptone water were done at both 4 degrees and 10 degrees centigrade. 50 and 100 parts or 100 micrograms of lactoferrin B was found to reduce the E. coli 157 by about 1 and 2 logs respectively within a day, depending on temperature. Higher the temperature, the more effective it was. When these studies were done in ground beef, again at 4 degrees and 10 degrees centigrade, same concentration, or the highest concentration, 100 micrograms of lactoferrin B, it reduced the E. coli 157 contamination by about 1 log within 3 to 5 days. So it did have an effect. And the reality of it is, when E. coli 157 is present in ground beef, it's usually at a very low level, usually at less than a log per gram. So lactoferrin B could have a benefit if low-level contamination of E. coli 157 is present. There are products on the market, and I give you an example here, something called Sausage Maker. You can go to the web and find this, which are fermented products that can have antimicrobial activity. In this particular example, it's a fermented product that's dried. It's a dairy-based product. It's whey, basically, that's in which an organism is grown, and it's a lactic acid bacteria that grows in the whey, and then it's dried as a powder, and then it can be added in sausage making, and it can help reduce pathogen contamination like controlled Staphylococcus aureus in the dried cured sausage making process. But there are other examples of this concept where lactic acid bacteria or something like Propionibacterium shermanii, which produces propionic acid, can be added to dairy whey and cultured, and then the whey is dried and it's added as a fermented dry whey product to bakery products such as bread. And this is, in quotes, a natural antimicrobial formulation because it doesn't have to be labeled as what the active ingredient is, which is propionic acid. It's simply labeled as cultured whey. Dairy whey has gotten to be quite expensive compared to what it used to be, and so there may be other opportunities for other waste products such as soy whey from making soy protein isolate may be another option in making such a substrate. So a few concluding comments. Levulinic acid plus sodium dodecyl sulfate at the appropriate concentrations and temperature can be an effective antimicrobial wash treatment to kill pathogens on a variety of foods without adverse consequences to the food. Alloisothiocyanate is naturally occurring or formed in horseradish and wasabi and mustard oil. It's highly bactericidal to E. coli 157 in certain foods like beef products. However, it does carry a relatively strong flavor and odor, and so that has to be addressed. And here in the U.S., there are regulatory hurdles. Frankly, the FDA has concerns about the safety of isothiocyanate. So in some countries this can be used, but not necessarily in the U.S. Using a combination of natural antimicrobial treatments to increase the efficacy and reduce the likelihood of the acquisition of pathogen resistance is wise. What I'm saying is I wouldn't rely solely on just one naturally occurring chemical, but to use combinations, because there's always questions that will be raised as to the likelihood that that pathogens with time and exposure would be developing some degree of resistance to these types of antimicrobials. So thank you for your interest and attention.